regulation, and doing so understanding the biological phenomena involved, and then showing you how you can identify those, how you can discover them using high-throughput sequencing. So the first part is going to be biology. And I have to tell you up front, I already told you, so I'm not a biologist. I learned those just as you learn, but probably in fewer courses. So if I'm saying something that might be outdated, please feel free to bring that up. I'm telling you what I know. I'm also giving you references for everything that I present, so that you can go back to the paper and you can look at the figure. You can read more information about the particular aspect of the lecture. You can email me at any time about those references or any further explanation about the lectures and so on. And if I speak too fast, my accent is too strong or anything, just stop me in the moment and I'll be able to hopefully to correct that. Okay, so back to this, the part one. The first part is going to be talking about the mechanisms of post-transcriptional gene regulation, and we're going to go through the operations that we place into these categories. And for the second part, I will give you some idea about how we can do analysis with next generation sequencing technology and a little bit about long reads. So how can we interrogate the transcriptome with sequencing data? Okay, so without further ado, some very brief and very general intro, so we know what the genome is. We're going to be talking about the genomes of eukaryotes in this class, and specifically we're going to be talking about the genomes of humans. So the genome is double-stranded. It is organized in 3D chromosomes. If we're looking across the genome, we see that there are genes. So we have genes located on the genome, about 20-something thousands of them, total genes, and a similar number, a little less of non-coded genes. And I think you heard about that on Monday. So if you're looking at a very high level, basically you can see the genes and you might imagine them as beads along a string, and we have genes on the forward strand and on the reverse strand. So everything here looks like a block represents a gene, but actually if we're going inside the gene, we see that the genes are interrupted, and they actually consist on the genome, of a sequence of blocks. Informative blocks, which we call exons, that are separated by non-informative, at least that's what we see from the purpose of this class, blocks that are called introns. So the exons are going to make it into the RNA, into the actual gene content. The introns are going to be spliced, are going to remain behind. So we'll be using this interrupted structure of the gene in order to go through the process and explain the process of gene expression, which is how a gene produces a functional protein, and that's what genes are for. That's why they are important. Again, we're on the genome. So a gene has a location on the genome. It has a start. It has an end. It has those blocks in the middle of exons and introns. The process of gene expression consists of two big steps and a few smaller intermediate steps. So the first one is transcription. So during transcription, the polymerase II makes a copy of the so-called coding strand of the gene, so the strand on which the gene is considered to be located. And basically, it replaces every base there for another base in this so-called mRNA transfer. One difference is that instead of T's, we have U's in the RNA. One disclaimer here, I'm probably going to be using U and T interchangeably. Just know that it's RNA and therefore it should be U, but nobody really uses U. So when we're talking about it. Okay, so that's the first step of transcription. We have a long molecule. This molecule is single-stranded, it's unstable, and it needs to go, for stabilization, it needs to go through a number of operations in order to become stable and then to go to the next stage to become a protein. So the first one is called capping at the 5' end. Then there's a cleavage and addition towards the 3' end, with the addition of 250 or so adenine base, a poly-A stretch, and that happens at the site that's in the neighborhood of a site that's called polyadenylation site. So you might see symbols here. So we have capping, we have polyadenylation, we have editing, which means changes to the underlying RNA molecule on a base by base at individual basis. That will be talking more about that. And the most important, perhaps for today's lecture and even for this process, is splicing. So the splicing consists in the removal of these intermediate sections, the introns, with the joining of adjacent exons to form the messenger RNA molecule. So all of this was happening in the nucleus. The mRNA now travels to the cytoplasm where it is translated. So again, this is the structure for protein coding gene, so that's gene expression. And the translation takes place not over the entire length, but rather starts at the methionine, at the codon that encodes for methionine and Athg, and stops as soon as, going 3 by 3, codon by codon, stops as soon as it encounters one of 3 stop codons. So that's TAG, TAA, and TGA. And I'm using T instead of U, so always replace that. So that will be the final protein, and there's the process of gene expression. I'm going to show you this so that you can see certain elements along the genome that represent binding sites that are important for facilitating the processes that we'll be talking about. So the post-transcriptional modifications that we'll be talking about are the following: the 5' capping, editing, splicing, polyadenylation, and then a process that is not per se post-transcriptional in the sense that it doesn't happen in the nucleus, it actually happens in the cytoplasm, and this nonsense mediated decay. But it still happens on the mRNA molecule. So I'll start with the capping. So what you have here on the right-hand side, you see that is the mRNA transcript. The mRNA transcript has was created by the incorporation of adenine cyclic cytosine bases, and in a 5' to 3' orientation. So it has a 5-phosphate group that is free at the 5' end, and it has a hydroxyl group that is free at the 3' end. And the capping here consists in the addition of a guanine triphosphate molecule at the 5' end of the pre-mRNA. So what happens here in the reaction is that the 5' to 3' GDP and the 5' to 3' nascent mRNA transcript interact. The reaction is catalyzed by the enzyme guanylyl transferase, and it releases a triphosphate to create a bond between a 5' facing and a 3' facing, between 2 5' facing molecules. Okay, so basically what you have now, you have a structure, 5' structure, that is distinct from the rest of the transcript, and the role of that is it allows the ribosome to recognize the start of the mRNA transcript, in addition to contributing to stability. So you're going to see many diagrams here. Here's how I read the diagrams. I look at everything that's there, and I'm trying to figure out what they do. So when in doubt, look at that and parse it, and to understand what's going on. Okay, so that was capping. The next operation is editing. And editing takes place when there's a change in the sequence of the RNA. So there's a change in the RNA that is not encoded in the genome. In human, we have by and large, we have two types of editing operations, and they are undertaken by 2 classes of proteins. So one of them, the first one, is the A to I, but that inozyme is read as a G. So this type of editing is mediated by the ADAR family of proteins. There's ADAR1, ADAR2, and ADAR3, and 3 is only specific to gray. Some properties of these is that they require a double-stranded substrate. So oftentimes you're going to see this happening. Actually, let me start with something else. So So this happens, the A to Y editing happens across the entire molecule, the entire pre-mRNA molecule. So that's the molecule that contains introns and in addition to exons. So most of the A to Y editing, in fact, happens in introns, and it requires a double substrate. Now, how can you get a double substrate? By having perhaps hybridization with another type of RNA, or more often, you're going to see the position of alu repeats, because the alus form a secondary structure that allows for the binding of the ADAR and the operation of the ADAR protein. It's not very specific, so you're typically going to see a cluster of edits that happen within a vicinity. So that's the first type of editing operation. The other one is C2U, or I'm going to refer to that as C2T. That's mediated by the ApoBet 1, 2, and 3 proteins. And that takes place on the mRNA. So that means that it can only happen in the exons. So it's on the spliced molecule. C2T also occurs in the DNA, but it's not the topic of today's discussion. These are the classes that happen in humans. In human, your questions about other types. What I can tell you for sure is that if you're going to other organisms, you might see different types of editing, and plants, for instance, have some types of editing where you can have small indels. One example, the textbook example of editing is shown here on the right. So we have the ApoB gene. The ApoB gene has 4,500 codons, so it codes for 4,500 amino acids. And there's editing of codon 2153 shown there, so that that's the codon CAA that codes for glutamine. However, that happens specifically in a given tissue. So let's compare two tissues. So in liver, there's no editing, and therefore the full protein is being translated, leading to a 4,500 amino acid protein with a function of transport of cholesterol in the blood. On the other hand, if we're looking at intestine, the molecule, the codon C in the CAA codon is edited to a U, which gives us a stop codon and produces a truncated protein that is only 2,100 amino acids long. This protein also has a very different function, and the function is the absorption of lipids from the intestine. So you can see how editing here actually contributed in a very significant way to creating two different protein products that had very significant differences in their function. However, if we're looking at RNA editing in general, the effects are usually not that strong. So rather they modulate perhaps the expression level. Many of those changes might actually be synonymous changes, so they might not lead to changes in the amino acid. Occasionally there are changes that produce or that inhibit splice sites, and those are probably some of the most significant. There are functional consequences, and inhibition of RNA editing for one reason or another, maybe usually because of defects in the ADAR proteins, have been associated with have had consequences for development and for some neurological disorders, for instance, epilepsy. So any questions about what I've said so far? Okay. So the third type of editing operation that I want to tell you about is splicing. So splicing consists in the removal of exons and joining of neighboring exons. Splicing is undertaken by the spliceosome, which is a complex of 5 so-called small nuclear ribonuclear proteins. I call them SNRMPs. I know they should be SNRPs. Okay, so those are U1, U2, U4, U5, U6. And that's happening along with the whole set of splicing auxiliary factors. There are around 80. We still don't know all of them, even though we have a fairly good imaging at this point. Some of those are listed here, U2AF1, U2AF2, and you will see some more in the following slides. These are so-called trans-actin elements. They bind specifically to sites within the mRNA, within the pre-mRNA in order to do their job. And the most important of those would be the consensus sites, the splice sites. This is the donor and the acceptor site. We call them donor and acceptor. So basically it's the 5' end of the intron and the 3' end of the intron, respectively. And you will see that in the vast majority of cases, the dinucleotides are G, D, or GU, and AG. We'll be talking about non-canonical splice sites in a couple of slides. Another important part for recognition by the spliceosome is a sequence at the branch point with a specific A, and the consensus sequence for the branch site is shown here. Y is a pyrimidine, R denotes a purine, and N is any nucleotide. So that's in your code. About the splicing process itself, so it takes place in two stages, two operations that are called trans-esterification reactions. So here's what's going on. So first the adenine at the branch site, we say that attacks the 5' end of the intron, the one in there, it detaches it, it cleaves it, and then it forms a bond. So that is preceded by this, and I should have said, by the binding of the U1 at the 5' end of the intron. So U1 binds at the 5' end, we have the attack, and the first trans-esterification reaction, which ends up with this structure, which is called the variant. That's number one. A number of SNRs, U2, U4, and U6 join to bring these sites close to each other in proximity, especially to form the lariat. And then U5 is also added at the 3' end of the intron. And the next trans-esterification reaction is where now the free end of the 5' previously 5' end of the exon, of the intron, so 3' end of the exon, now attacks the guanine at the end, 3' end of the intron, forming a reaction, a bond, and freeing this structure that is called the lariat. Okay, so two trans-esterification relations that produce a cleavage and a joining. And this is undertaken by the spliceosome. So this is the basic process of splicing. Questions so far? And you saw the sequences GU and AG. So those are called the consensus or the splice, consensus splice signals or consensus splice sites. We call them canonical. We call the GU AG the canonical splice sites. They occur in, so the major spliceosome, the one that we talked about on the previous slide, undertakes splicing in about 99% of the cases, or about 99% of the exons, maybe, of the introns, maybe a little bit more. But there's another component, a spliceosome component, or another variation, which is called the minor spliceosome, which is responsible for the remaining portion, less than 1%. So the minor spliceosome resembles the major spliceosome in the sense that it has a similar collection of proteins and a similar complex. However, now U1, U2, U4, and U6 are all replaced, and only U5 is in common to the two spliceosomes. So they are similar in structure, but they recognize different motifs. The motifs that they recognize now most frequently include ATAC. So that's the most frequent for the minor spliceosome. But you will also see GTAG, ATAG, and a number of other variations. So if you're actually looking at the gen code annotation, which truth be told, we cannot guarantee that it's really that it's entirely true. But you will see that there are many variations that you can see just on a case-by-case basis, accounting for those at most 1% of the splice sites. Now, to make things a little bit more complicated, it is possible for the major spliceosome to also recognize GCAG types of signals. And it is also possible for the minor spliceosome to recognize GP and G types of signals. So just because this consensus splice site looks a certain way, we cannot guarantee that we can tell which of the spliceosomes produced is undertaking the splicing. So just keep in mind that sometimes if your alignment program is not doing things properly, you're running STAR or you're running another alignment program that is supposed to be recognizing canonical splice sites, it might not be good at identifying non-canonical splice sites. Okay, so next, splicing. So in general, we assume that splicing takes place where it needs to take place. And the genes are expressed where they need to be expressed. Genes, so not all genes are expressed in all tissues and in all cell types. So we know that. Not all of the splice sites are going to be used everywhere, but we'll talk about alternative splicing in a couple of slides. There are also many loci on the genome where splicing that look like exons, where splicing could occur, but splicing is actually repressed because it would be deleterious perhaps to the organism. So there's a repressive mechanism that might be keeping some exons from being expressed, either in the wrong tissue or just being expressed anywhere. And we call those cryptic splicing. So that's what you would not expect to see in a regular cell. There are a number of proteins that are undertaking this repression. Two that are very well known are H and RNPC. And another one is actually the U1 SNRP. So H and RNPC usually appears, that's the C there, and appears as a tetradimer, and it binds close to the, or even at the branch site. By binding to the branch site, it prohibits the binding of other proteins and other SNRPs, particularly to affiliated factor, associated factor, and therefore inhibits the splicing of a cryptic exon. So it's acting from within an intron. On the other hand, U1 would inhibit the splicing of a potential of a cryptic exon from within the exon. So the binding sites for U1 would be is this 4 base motif that would occur within the potential exon. And it prevents the accumulation of U1 and the rest of the spliceosome. So they should be repressed. And bad things can happen if they are not. How can they not be repressed? Because of geneticists. So let's think about that. So let's say that something happens where there is a mutation that changes the protein in a way that changes its binding, or the efficacy of binding. So, or the availability of a substrate, and that can happen. There are a number of diseases. There's frontotemporal dementia, ALS, accumulation of these cryptic exons. But perhaps of more interest to this class is that there can be, on a gene-by-gene basis, mutations that abolish these particular binding sites that are protected. So I'm going to show you 4 examples, and again, for this one you have the reference here. So I'm going to show you 4 examples of diseases where we have mutations in a gene that lead to the creation of a cryptic exon, which in turn leads to the disease. Okay, so the first example that is shown here, there's a little alpha, is exactly the example that I showed you before. So One particular case is that of ARU transpose elements. So the ARU elements, especially when in antisense to the gene, have a sequence that makes them, that gives them the proclivity to create new splice sites. So it is fairly easy for an ARU with just a few changes to the sequence, small changes to the sequence to become exonized and incorporated into a protein. Therefore, HNRNPC works as a global regulator of repression of this ARU incorporation. So the first example that I'm going to tell you is, so this alpha shows what I mentioned above. So this tetradimer of HNRNPC would bind upstream of a cryptic exon that would result from an ARU and would block the binding of Q2AF, so block the binding of the spliceosome that would lead to the expression of the exon. In the disease that is called, there is a very long term, hyperphenylalanemia. At the gene, the PTS gene, it's impossible for me to remember the name. So it's PTS gene. There's a mutation that removes a portion of the sequence here that contains the binding site for the HNRNPC protein. By abolishing that, that creates the space for the spliceosome to add and to allow the incorporation of the ARU-derived exon. And as I said, the result is the disease. So that's the first example. The second example shown with beta here. So again, we are in an otherwise deep intronic region between these two exons. So in the second case, at the gene that occurs at the gene ATM, ataxia telangiectasia. Why do they make names so difficult? ATM. That is much easier. I'm trying to remember what M is. Well, at the ATM gene, so a four base motif, this motif, in an otherwise cryptic exon are being removed. That happens to be the binding site of the repressing repressing U1 slur. And with it, the repressive mechanism is abolished, and the exon becomes included into the gene to cause ataxia telangiectasia. Kirby, if you can help me out with these names, because I really can't get them. Ataxia telangiectasia.
Letters work fine.
Okay. Yeah. The third case here appears at the CFTR gene. So you know that one because that's related to cystic fibrosis. So it's simply a mutation that creates a new splice site, a GU splice site, a donor site, and that promotes the incorporation of an exon, of a cryptic exon. So we already said what the disease is. And the fourth example that I'm showing you here happens in the BRCA2 gene. So the BRCA2 gene, an otherwise known and canonical site, AG. So the A in AG, A is mutated to a G. The exon is no longer recognized. Instead, a cryptic exon that is upstream of it is being recognized and being incorporated into the gene. So I don't need to tell you that BRCA2 is mutations in BRCA2 have been associated, including this one, with a number of diseases: breast, cervical cancer, some prostate cancer, somewhere in that area. So these are just 4 examples, and there are probably more, but we don't know all of them. The good thing is that with next generation, or rather I'm going to call it high-throughput sequencing, because now we have long reads as well, we can actually interrogate these and we can look at them based on biopsy samples, and we can characterize cryptic splicing genome-wide. So that's a reference to one paper that we did, and there's a reference to the PDP-43 repression leading to accumulation but I don't know more about that.
So kind of jumping off that, I know in like lots of cancers, there's this global hypomethylation, especially the allo elements. Does that result in some of these cryptic exons being
Being incorporated? Could be. Yeah, could be. So I do know that the methylation plays a part in is one of the repressive mechanisms, but there are several others for transphosphoramines. So there are variations to this splicing. So whenever you think that something is clear and clean, it turns out to not be that way. So you recognize these. We have the gene. Splicing takes place. It puts the exons together. It forms an mRNA, and we have a protein, right? And that's all good, except that that is not always the case. So that may happen in some tissues under some conditions. However, we move on to another tissue, another cell type, and then we see that different combinations of exons of the gene exon can be used to produce a different variant of the transcript and potentially of the resulting protein. So we call the process alternative splicing. The results of this, to allow for this variation, we refer to them as transcript variants, or as transcripts, or as splice variants, something along those lines. So those terms are used interchangeably. What do these variations look like? The one example that I'm showing you here simply shows two transcript variants where the first one contains all of the gene's exons, exon 1, 2, and 3, and the second one shows only exons 1 and 3. So we call that exon skipping because the exon in the middle, exon number 2, is present in some transcripts of the gene and is missing from others. But alternative splicing can actually take other forms, and these are also a number of just a subset of them. So people like to be very clear about what is being extracted and to refer to that portion and see, is it an exon, is it a portion of an exon, and so on. So this is a very clean representation of the so-called canonical primitive splicing variation that one would expect. You see at the top exon skipping, which is what we saw on the previous slide. Then we can have mutually exclusive exons when different transcripts would use one, but never two of two exons. We can have alternative 3', and this is a little bit confusing here. So I'm calling it 5', 3', a 3' exon end, but some tools and some textbooks will refer to that as the 5' splice site. So alternative 5' splice site. Because there's the 3' end of the exon, but it's the 5' splice site. It refers to the exon. Okay? And we can have that at the other end of the exon, we can have intron retention, so where the intron is not spliced out. And two categories here that are not splicing per se. So the first one is the alternative gene start, or alternative promoter, and alternative Fourier derivation. And we'll talk about each of those a bit. But as I was saying, it's rarely as clear and as clean as we would like and as we represent it. So these are view from the UCSC genome browser for the BRCA1 gene, where each one of these shows you a splice variant. The blue ones are predicted. For the black ones, we have very high confidence and perhaps direct molecular evidence. And these are exons. Vertical bars are exons. So you can see here that this one skips all of these exons. So you can have multiple exons being skipped. You can have this early termination. You can have this being skipped. You can have a variation here entirely, both of these. So entire exon with a variation and the next exon are being skipped here and so on. So clearly splicing is much more complicated than the few patterns that I showed you on the previous slide. So how is this happening? Well, we learned so far that, and that was the basic view of splicing, is that what is important here is that you have a GU motif, and actually it's a little bit more than GU, because U1 recognizes a slightly longer motif sequence there. So we have the GU, we have the AGU on the other end, again a slightly larger vicinity that is being recognized, and then we have the branch site. It turns out that for alternative splicing, when alternative splicing happens, and that happens at more than 90% of the human genes, for instance, then that process gets much more complicated. So if you're looking at all this complicated figure, basically this is what you have to think. So we have, in addition to the basic apparatus that we talked about, we have elements that help with splicing, activating splicing, and we have elements that inhibit splicing. And we have trans factors, which are proteins, and we have cis factors. So the cis factors are the binding sites of those proteins to the mRNA. Okay, so everything here is coded that way. So let's look along the genome at the cis sites. Okay, we can have, if they are located in the exon, we're going to call them exonic splicing enhancer, which helps with activating the splicing, and exonic splicing silencer. So that's what these ESC and ESS represent. But we can have binding sites, so we can have proteins acting on sites within the intron. So then we're going to call them by the intron. So intronic splicing enhancer, and intronic splicing silencer. Okay? And for the proteins, we really have them as splicing activators or splicing silencers or repressors or inhibitors. So that should make it a little bit easier. And all of these lines here, when you see an arrow, that means that it helps activate. When you see a bar, I'm sure that you're familiar with that. That means that it represses. So that's how you read. That's how you read these. So over the next few slides, we're going to go through what are the factors, the trans factors, and what we know about these motifs and how they work together. And for this, by the way, so one of the suggested readings is actually a paper by Luca Carteni and Adrian Kreiner. It's an old paper, but it's still, I think, one of the best papers that I have ever written. And some of these were taken from that paper. So you can go and you can look at those, at some of those. Okay, so let's talk about activation, splicing activation first. So promoting the splicing of alternatively spliced exons or variations. By and large, we think that happens in two ways. So actually, let me say a little bit. So first about the splicing factors, splicing activators. There's this class of proteins that are called SR proteins. And they're splicing enhancers. They have one or two RNA recognition motifs, which have them bind to the genome, to the mRNA. And they have an arginine serine-rich terminal domain. There are a number of such factors. Some of them are listed here. And we have databases where if you want to find if there's a potential splice, or potential binding sites for those motifs, you can put in your sequence and you can search for motifs. Early on, there's one that was called DSC Finder by the same people who authored the paper. There's now a much larger database, Athg, which is not only for splicing factors per se, it's really more for RNA binding proteins, but it is very rich and it's based on a richness of data. So I would recommend this database if you ever want to study splicing regulation and you want to look for those motifs. So the SR proteins are the splicing enhancers. What are then the mechanisms? So again, when you see something as complicated as this, it looks complicated to me. I'm trying to look at the components and put them in the context of what I know. So we have exon. We have U1. We have binding at the 5' splice site. We have U2 SNR with the auxiliary factor U2AF and so on at the branch site and the okay. So one way that this can be enhanced is through a recruiting function. So we have an SR protein that binds to an exonic splicing enhancer with its RRM, RNA recognition motif, and that has an RS domain through which it actually exercises its enhancing function, either by through either direct interaction or indirect interaction. So, for instance, by directly interacting with the U1 at the 3' end of the exon, or by interacting with a U2AF factor, or indirectly interacting with the splicing co-activators such as SRM160. So basically it binds to an ESC and through its RS domain, it performs its function. The second way it can perform the function is actually in an antagonist way. So we're talking here about competition between this activator and the repressor where the activator wins. So that's all that is here. We have an exon, a splicing enhancer element here, and also splicing silencer somewhere close to it. And the RNA recognition motif, the protein with its RNA recognition motif binds to the ESC and inhibits and precludes the inhibitor from performing its repressive function. So the first one is directly active, the second one is it prevents the inhibition by another element. And there are probably other ways. How about inhibition or silencing? So these are some of the mechanisms. The first one is what we saw on the other side. So we have the direct competition. We have sites within an exon for enhancing the splicing and for silencing the splicing, and in this case, the silencer, the silencing mechanism wins. Okay, so it's direct competition for one. And the exon, in that case, the exon is being skipped. The second one is a mechanism that's fairly rare. Yeah. Oh no, okay. Yeah, so it's fairly rare. So that's called exon looping. And that happens when, let's say, this is the exon that is alternatively spliced. So we have a dimer that binds to, that has binding sites on both sides, and relatively close to the ends of the exon. And the two components dimerize, and they bring the two strands together. Therefore, putting the exon in a configuration that is not accessible to the spliceosome. So that's set aside. The spliceosome goes and connects the adjacent, the neighboring exon, skipping this exon. Okay, so that's another mechanism. And the third one is a variation on the first one, where it's nucleation and cooperative binding. So basically, there are multiple sites, and multiple sites in addition to some enhancer, and then we have binding to a high affinity site by a silencer, and then nucleation, so cooperative binding to a nearby site, a nearby site, a nearby site, until it reaches a point where it produces competition to the actual enhancer. So it's really just a variation on the first one. I'm showing you because that's what it looks like. As I said, I believe that biology is probably more complicated.
What makes the exon looping rare compared to the others?
Because you need to have binding sites of a protein that forms a dimer. Yeah, that's all. And you know, if my memory was any better, I would be able to tell you it's something. I can look for that. I believe that it's happening in a disease. I read something even recently of an example, but I can't remember. I just can't remember. Not very frequent though.
Yeah.
Okay, so alternative splicing in disease. Pretty much almost any disease that one is looking at, you have to wonder whether there are variations in splicing. Some of them are global in the sense that there might be changes in the ratio or the expression level, perhaps, or the splice variation of a splicing regulator. So those would affect splicing globally, and that's what you start seeing. That's what you see in pretty much every cancer. But for this particular group, so for our field, we're really interested in those gene-based point mutations that could lead to erroneous splicing, and aberrant splicing of that particular gene, and in turn lead to disease. And I'm going to focus on those. And by the way, those are more than we would expect. So 15% of point mutations in a database, I think it was AG, Human Genome Mutation Database. So 50% of the point mutation resulting in genetic disease actually lead to splicing defects. And then then to clarify, when I'm talking about aberrant splicing, so what can potentially coexist? Well, we can have the wrong variant expressed in a cell, potentially with the expression of a cryptic exon, or we can even have the right variants but expressed in the wrong ratio. So these are some of the things to keep in mind. So here are some examples. I'm going to start with this one. It's fibroblast growth factor receptor 2. And its splice variation, it's the role of that splice variation in prostate cancer progression. So what you see here is epithelial cell model and the mesenchymal cell model. So in a regular cell in the epithelial stage, let me put it. So we have two mutually exclusive, an example of a mutually exclusive exon use. We have two constitutive exons, upstream and downstream, called here. And then we have 2 and 4 perhaps. And then we have two mutually exclusive exons that you see in the middle. Let's call them 3B and 3C. And then there's a complex regulatory system here. So if you see here, there's a so-called intronic splicing activator repressor, which does a dual job. So on the one hand, it represses 3C, but it activates, it promotes the inclusion of 3B. And then we have at least 2 splicing silencers that are upstream of 3B that would silence, so would compete with the activation from ISIR. So it's a fairly complex mechanism that takes place there. So where is the aberration coming from? In the regular, in the expected case, exon 3B is included, and 3C may or may not be included, but it's included at very low levels. So exon 3B is primarily expressed in epithelial stage. As it progresses towards the mesenchymal stage, exon 3B is being silenced, and only exons 3C is being expressed. So that's it. It's just the expression of a different exon in a mutually exclusive exon combination. Again, references here. If you want to read beautiful paper, if you want to read more about this entire process, and here I'll read papers after that. So questions about that? So the next example that I want to show you, which you've probably heard about in some of your other classes, is the example in the spinal muscular atrophy. So there are 2 genes, SMN1 and SMN, spinal motor neuron 1 and spinal motor neuron 2. They are identical except for a small change in exon 7, where at position 6, the C in SMN1, its corresponding position is U in SMN2. So spinal muscular atrophy occurs when both copies of SMN1 are inactive in an individual. Okay? So let me tell you a little bit more about these genes. So that mutation happens to be in an exonic splicing enhancer that would promote the inclusion of that exon. So by abolishing the ESC, the inclusion of this exon becomes very inefficient, and only about 20% of the outcome of the gene of the protein is being produced. You might contain the entirety of the gene. You might say, but what happens to the protein when you're skipping that? Well, that turns out to be non-functional and it gets degraded. So that's a defective protein. So now let's dissect that. In a normal individual, okay, we have both SMN1 and SMN2 active. SMN1 will produce 100% of its output, and SMN2, because it has the mutation and exonic splicing enhancer, will produce about 20% of the functional protein and 50% of its defective SMN delta 7 protein that doesn't do anything, at least good for us. Now, and as long as at least one copy of SMN1 is active, is correct, then there are no problems. However, if both copies are inactive, then the only gene that can produce the protein is SMN2, and it does so inefficiently at only 20% of its at the level of output. So that's the basis. That's the basis for which the disease. There's a basis for the spinal muscular atrophy disease. So is that clear? I don't know how disease progresses and how it happens over time, but this is the mechanism. Okay. So the next two editing operations that I want to talk, and I'll try to make it short, will be alternative polyaddenylation. I put the alternative because there are multiple possibilities, and then we'll be talking about non-sensitive. So polyaddenylation, it's one of the operations that happens on the pmRNA molecule that takes place as cleavage of the pmRNA transcript at a site that is recognizable via a polyadenylation site sequence, which is fairly well conserved, and with the addition of a polyene tail. Just like with every process, we have both trans factors and cis factors. So the trans factors here are two proteins, the CPSF, cleavage, I have to write them down because I can't remember that. Cleavage and polyadenylation specificity factor, so it's a CPSF there, which binds to a polyadenylation site sequence located about 10 to 30 nucleotides upstream of the actual site of cleavage. So that's one of them. And the site sequence there is written as AAU, AAA. In fact, it can recognize many more sequences, and there was a paper by Jan, I think I mentioned it on the previous slide, that there are 13 motifs that account for about 75% of all these instances. So 25% still have a sequence that looks nothing like 30. In biology, everything, few things are precise, very, very precise, because redundancy is necessary for the system to function and to be maintained. The second part, which is sometimes necessary, is the binding of the CPSF, the cleavage stimulator factor, that happens at the site that is U or GU reach. It's 20 to 40 nucleotides downstream for the actual cleavage site. And now when I'm talking about the cleavage site, it's not a precise site. It's actually, it was identified to be within a 24 base pair window. So anywhere within that 24 base pair window, you can have the end of the transcript. So that's as far as the mechanism, or rather the components of the mechanism are involved. I want to show you how those manifest themselves though. So we can have, these are all transcripts and the 3' ends of the transcript. So we can have a single polyadenylation site per gene, or maybe for a transcript. And that's the case that I'm showing you here. So that's the last exon, all good. We can also have multiple potential polyadenylation sites within a very large last exon. So any of them might be chosen. And as you can imagine, choosing a different one would actually affect what sequence is available for binding of microRNAs and other things, which I'm sure you talked about on Monday. Also stability, the sequence is important for stability, and so on. So in this case, basically, choosing one of these three can lead, choosing any one would lead to a different transcript, potentially slightly longer proteins. And then we can also have situations which are very difficult parentheses to identify with the next generation sequencing. So we can have a polyadenylation site that is located actually in the middle of an internal exon, just like it's shown here. So it produced this molecule here, like a truncated version of the normal molecule. We can have a polyadenylation site that's intronic, and the way it manifests itself, and you're going to see it in RNA exon and goes through in the intron until it finds a polydenylation site. That happens. And they are particularly difficult to identify. And then we can also have the selection, there can be an exon skip, and on the previous slide, we can even have something where we have this completely distinct, non-overlapping exons as the last exon of the gene. So there's a lot of variation that can be created by having different gene ends in the polydenylation sites. And as I was saying, so that sequence is fairly degenerate. Okay. Questions about that? Yeah.
So just to make sure sort of the sequence of things, the polydenylation site is really a pair of sites, or could be a pair of sites, one being the denylation factor binding site, the other being the fetus stimulating factor binding site, right?
Well, so the denylation site is only the first one, the AAUAAA is the first one. The second one might or might not be present. So it's only sometimes present.
Is the past always present?
As far as I know. Yeah. So something is.
But if it's not present, but like say you have 3 sites, if you hit the earliest one, it doesn't have a cleavage, the mRNA is still being cleaved before the poly detail is added.
It has to be cleaved because that's where the polydenylation is. So where it's cleaved, that's where it's not.
Got it.
Okay, so the last type of editing, even though it's really not editing operation, it's a surveillance mechanism. It's non-sensitive decay. And by the way, that's another one of the, why did I not say that? Another mechanism by which exons that are incorporated can be identified with the decay. So watch to see how it's happening. So it's a surveillance mechanism that identifies the incorporation of exons that contain a premature termination codon and therefore will cause a mutation of the protein which acts to degrade the transcript. So let me show you what that looks like. This is all happening in the cytoplasm. So not in the nucleus. And before being considered for translation. So let's say that we have a gene with 6 exons. If there's no PTC, premature termination codon, what we expect to see is 100% of the mRNA, whatever the gene expression level is, and 100% of the protein, or the protein will incorporate everything that's, so these are only the coding sequence, or the exons that form the coding sequence. So it's a full protein, full mRNA. So now let's think that if we have nonsense mutation and premature termination codon, we would predict to have 100% of the mRNA because it's still being produced, right? And we would expect to have maybe 100% of protein that is truncated at the point of the PTC. However, that's not true. If we're looking at the output, we're going to see that there's a very small amount of mRNA from the gene and 0 protein. So that's because of this degradation mechanism. Now, the location of this PTC within the genes, or rather the transcript, is very important. And that gets me to the rules here. So the first rule is that is the 5' to the 3' rule. So the more 3', the less the effect. And conversely, the more 5', the more towards the beginning of the gene, the bigger the effect. So for instance, if the PTC is in the first exon, you might only see 3% of the output. Most of the gene has been degraded. The gene output was degraded. If it's in the second, you might see a little bit more of the output. Now, there's also a threshold there, and that's the exon junction rule. If the PTC is located in the last exon, or within 50 bases of the last exon-exon junction, it does not have any effect. So there's no degradation. And you might see why, because it's related to the exon junction here. I'll show you on the next slide why. So the effect is strongest if it happens toward the beginning of the protein, and it's weakest towards the end, with no effect if it happens within this last part. so past the last exon-exon junction or within close proximity to that. So why is that? So here's a possible mechanism. We know that it's true, but this is the reference that I gave you. It's fairly old. I know there's a more recent review. So this is what happens. So we start with a gene, or with a, in this case with a transcript, pre-mRNA splicing will produce an mRNA. But upon splicing, so the spliceosome deposits something that we call an exon junction complex on every exon-exon junction. So that's what you see this accumulation at the top. The molecule goes from the nucleus into the cytoplasm, and it should undergo translation, recognition by the ribosome, and proceeding with translation. So, on the regular wild-type molecule, we don't have a premature termination codon. There's an initial level, an initial step of translation. So the first time the ribosome goes through. So the first time the ribosome goes through, it displaces all the EJCs. So it displaces all the exon junction complexes. So at subsequent rounds of translation, there's no impediment, and the full peptide is being formed, the full protein is being formed. Because the ribosome managed to complete its way through the mRNA. So that's what happens if we don't have a PTC. If we do have a premature termination codon somewhere not in the last exon and not too close to the last exon-exon junction, you might already guess now. So the first round of translation, during the first round of translation, the ribosome displaces all these exon junction complexes, but only the ones that it sees. It stops as soon as it sees a PTC. So we know that there's at least one exon junction complex that cannot be removed. Okay? So there's one there that shouldn't be. So at the next, in subsequent rounds, the ribosome might cross, but it cannot produce the entire protein. And there's actually an interaction between the post-termination complex and a set of proteins that are called upframe shift proteins. So these are the surveyors that look for the presence of exon junction complexes. So no PTC means that exon junction complexes have all been removed and translation can proceed unimpeded. PTC is present at any exon-exon junction other than the last one. Therefore, not all of them will be removed, and there's a recognition by the UPF proteins that leads to mRNA degradation. Okay, cool. So
the ones that are within 50 bases of that, are they just shielded by the exon junction complex?
Yeah.
And so just ignored?
Yeah, so it's the proximity, because nothing is very, very specific perhaps. Yeah. Okay. So now I'm gonna go to the part, and I know I don't have much, so I will go through that pretty fast. However, and maybe that's what I wanna start with. So right at the end of it might seem self-serving, but it's in different way from what you think. Okay, so I'm teaching 2 Coursera courses. Okay, one of them is an introductory course that is called Online Tools for Genomic Data Science. So if you don't have any background in bioinformatics and you want to learn how to use Linux, how to use Unix, other things, how to do a basic variant code analysis, how to run SAM tools and things like that, alignment and the basic RNA, that would be a good course. That's also available, so it's from Coursera. Coursera is also available from the J2 forum. The second one is a much more recent one. And it's an intermediate course, so it's more like if you need to do a type of analysis. So it's an up-to-date type of RNA-seq analysis, for example, sequencing data, alternative splicing analysis, and it also has this type of analysis for long read. Now, both of them can be accessed without cost if you're not taking the exams. And for the second one, so the first one is definitely available from the portal. I think you can do the exam out without paying because it's in marketing. The second one, I put the scripts outside of the part with the test. So you can have the scripts and you can use the scripts even if you're not taking the exams and therefore you don't have to pay for that. So you can do it that way. So I want to have those there because I used to teach the RNA-seq during one of the classes and I'm no longer teaching that. So if you need that, that's a reference for you. So let me go at some high level through this, and you have the slides. So I want to touch on two types of analysis. I guess 10 minutes each. So let me see how I put them 10 minutes each. First one is with next generation sequencing, which we slowly transition to second generation sequencing. For RNA-seq, I'm not going to tell you, you have there why RNA-seq is better than microarrays. Well, it gives you the sequence. So you don't have just the intensity of the probe, it tells you the actual sequence. You can do very important things with the sequences. You can tell if you have a real specific expression. You can tell if you have mutations. You can tell anything that happened there. You can tell if you have fusions, and so on. You also have a much wider range of expression levels now, so you can be more precise with the expression. What's the problem there, though? The problem is that it tells you about the gene in small bits. So, second generation sequences, the sequencing produces bits that are only about 150 base pairs long. So if you want to put together the entire transcript, you have to put many of them together. And it turns out that it's a very complicated process because you can have multiple variants expressed in the same cell, so now you don't know which one goes with which. We have ways to do that, but the precision is still fairly inaccurate procedure. So a lot that we can do approximately, but it's good enough, and that's pretty much how we use them now. So that's the process. We start from RNA. That's what you're probably going to be doing in the lab. We start from RNA. That could be total RNA, or it could be polyadenylated RNA, and that matters, okay? Because polyadenylated RNA is going to include your coding fraction and also some link RNAs that are polyadenylated. But if there's no poly-A, then you need a total RNA. If you have degraded RNA, from a biopsy tissue that is paraffin embedded, you're not going to have a poly-A, you're not going to have a full transcript attached to the poly-A. So then you're going to do a total RNA. Okay, so that's the RNA. This RNA, there are two ways of handling that. One way, what used to be where you create an entire, you clone that to a cDNA, and then you chop the cDNA and amplify that. The way we're doing it now, turns out that it gives us better coverage, is you chop the RNA, and every piece, every fragment, which is about 500 bases, gets cloned, create a cDNA, and that, if needed, amplified before sequencing. So two procedures, you get more uniform coverage with this random fragmentation of the RNA. So you have these fragments, which potentially are amplified if you don't have enough RNA. And now sequencing happens. It might happen from just one end, and we call it single end, or it can happen simultaneously from both ends. So you can have paired them. Okay, so now imagine you have a fragment of 500, 150 this way, 150 this way. That 500 is an average. You might have shorter ones, these two look like that. We take these reads, so you go in with your samples, you create the libraries, the appropriate libraries, check with the sequencing port, and you have the data now. There's a pipeline. Okay, so first you map the list to the genome because you want to see what genes they came from, with an allocation of the genome. Second, we want to make sense of what the expression level of the genes would be, and we want to see any splice variation. So we do that during a process that's called transcript. So it's alignment and assembly. So transcript assembly. We can redo the quantification then with something that also does differential expression. But once we have that, in a typical experiment, you're not going to be doing an experiment on just one condition. You're going to say, I'm knocking off this protein, and I want to see, when I knock this off, what happens to the cell in general, or what happens to the tissue in general, right? So most of your experiments are going to be two conditions, maybe more conditions. One, that's the control.
wild type or whatever it is, untreated, and one that's treated or case or disease, whatever it is. So you're going to have two conditions. You need to have multiple replicas, and I'll tell you on one of the slides for each of the conditions. And in the end, after you look at every sample and you do alignment and assembly, and you come up with your database, your expressed genes and expressed splice variants there, you do the differential expression, and if you wish, differential splicing. So there are tools to do all of these. So I'll point you to that. A few things about experiment designs. Experiment design. Never have fewer than 3 replicas per condition. Does not allow for, you need to have that because the model needs to estimate the distribution and the biological variability in the negative binomial. Usually it's a negative binomial model. So at least 3 biological replicas per condition. Biological replicas doesn't mean that you went to your biology or whatever it is, and you created your library, and now you split that library into 3 things. It's not, so it has to be taken independently. Depth. So most of the experiments that you're going to need will be poly-A selection, because usually the phenotype driving protein coding genes are the ones that are phenotype driving. So very rarely is a link RNA related mechanism that is at play. So poly-A is very reliable, is standardized, so generally no problems there. If you want to just do differential gene expression, something very quick and dirty, you map to the reference annotation, 20 to 40 million reads. I know people are trying to squeeze as few reads, but sequencing is cheap. When you see 20 to 40 million reads, just know that only about 75% of those are actually going to map to your exons, even in poly-A selection. Because there's a fair amount of noise that comes from the not yet spliced exon introns. It's a biological process. Turns out that splicing and transcription are happening at the same time, so it might not have done. What we usually recommend, though, is this option here. 40 to 60 million paired N reads, okay? Because that allows one to actually do the transcript assembly and to be much more precise about the set of genes that is being expressed. If you only have this here, you can't do assembly. What you do is you map them and you see what genes seem to have a number of reads. But I can tell you that many of them will go to the wrong genes. There are many gene families, okay? And there's also a certain amount of noise. So strongly recommended that this is the option you use. These are the points I'm recommending to you. And go through the process of mapping, assembly, and then differential expression. If you are really interested in alternative splicing, you're studying a condition like ALS that is totally related to splicing, or you want to investigate that kind of space, you need at least 80 million high quality reads. I would say more like 120. In the study that I've done, use even more than that. Don't go to 300 million, it's too much. You start seeing noise, there are other issues. But somewhere in the vicinity of 80 to 150 million reads, that's what you need to look at splicing, to identify all the splicing variants for the ones that make sense. Now, if you have total RNA, as I said, comes from a biopsy that has been biopsy embedded, it comes from a mammoth hair that has been extracted from a frog someplace, something of that sort, Neanderthal hair, and so on. So that's RNA depleted because 90% of the RNA will be ribosomal RNA, so that needs to be taken aside. So there are ways to build the library, but you're going to need many more reads. You're going to need 3 or 4 times more reads, and here's why. Now, not only do you have the expression of the exons, but now total RNA includes the entire transcript, so it's in the process of being spliced. It hasn't been like 90% spliced. No, you're going to find, so there will be a significant amount of reads from the introns. Introns are long, so you're losing a lot of reads here on the introns, which are part of the pre-mRNA, right? Because the total RNA contains what? It contains the pre-mRNA. In in addition to the mRNA. So RNA in all of its stages. So careful with this one, you need much more. Many people want to do a survey of the non-coding RNA, and then they will be going for this one, for total RNA. Now that with poly-A selection, you actually get a large portion of the link RNAs and some other non-coding RNAs. So think about it, that might still be your best choice. This is the pipeline for short reads. Quality check, then mapping the list to the genome, assembling. Strongly recommending assembling because that looks at reads that can be grouped on the genome within gene buckets, okay? And they are very accurate at identifying potential artifacts that come from paralogs in gene families that should not be expressed. So that's there, and then B seek to do a comparison, differential comparison, and then there are tools to do differential splicing comparisons. Again, I'm going to refer you to the Coursera for practical recipes and for scripts. And I'm going to skip over this. Okay, a few words about transcriptomics with long RNA sequencing. So basically, the differences here. First of all, they have the same advantage. They are sequences, so they will have many of the advantages of the RNA-seq technology. You see the sequence. You can identify mutations there. However, keep in mind that the sequencing error rate here is going to be much higher than for short reads. Okay, so if you have many copies, yeah, you can identify mutations, but it's not as reliable as with short reads. The main benefit is that you can actually have end-to-end sequences. So they are oftentimes, most of them are more than 1 kilobase, maybe not most, but enough of them will be more than 1 kilobase long. The average size of an mRNA is 2,000 bases. We know some of them are much longer. But what that means is that these reads are likely to cover perhaps your entire transcript for a very large portion of it. They suddenly will capture the portion of the 3' end. Most technologies will capture the 3' end and perhaps leave something at the 5' end that's incomplete. But you will see some partial ones that it really matters how you build in the libraries as well. So that's the main benefit, that you don't need to do assembly. You don't need to put together all those pieces to say, this is splice variant 1, but I also have splice variant 2 that is skipping this exon or is starting an exon 3 and so on. So it's much more reliable from that point of view. I'm showing you here with short reads, you have to, for that gene at the top, in black you see the constitutive exons or portions of exons, in red you see the parts that are alternatively spliced. And just for the purpose of illustration here, let's say that we have 3 transcripts for this gene. You can probably see here that we have one skipped exon, we have another skipped exon here, and we have an alternative 5' end. And those are 3 transcripts, but we can see how this can be combined in many other ways. Some short read data would look like this. What I'm showing you here is a spliced read. And I should have shown you that one particular splice, but one particular slide. But let me ask you to just imagine this. So you have an RNA sequence, and now you broke the sequence and you obtain into fragments and you obtain a read. A read is 150 bases. Your mRNA is 2,000 bases. That read can be anywhere in the sequence, which means it can be from the middle of an exon. So it can all fit within an exon. An exon is about 150, 160 bases in the middle. So it can fit all in an exon. And we call that an unspliced read. Okay? Or it can be one of those unlucky ones, but not so few, that span the boundary between 2 consecutive exons. okay? So when they spend the boundary between two exons, think about when you map them to the genome. So there's the genome, there's one exon, there's another exon. A portion is going to map at the end of this exon, and the next portion is going to map at the beginning of the next exon. So what you have there is something that is called the spliced read. And if you're paying attention to that, what that tells us, that gives us two pieces of information. Number one, it tells us, oh, it fell here, it came from an RNA, that means that this is where there's a gene, there's a piece of gene, okay? It also tells us where the ends, where the intron is, because it jumps over the intron. So it tells us where the intron is located on the genome. So we call those spliced reads. And that's what I'm showing you here. So the short reads, these are the short reads. In yellow would be the unspliced reads. They fall squarely in the middle or the sides of an exon, but they don't need to be split into exons. And then we have spliced reads, where a portion matches at the end of one exon, but if the mRNA, it came from this one, then it's going to jump all the way here for the next match. Okay? So, to process these short reads, as I was saying, we have to figure out which one goes with which one, and so on. And using the quantities, how much we see the levels of the depth on different exons to deconvolute. It's a deconvolution problem. When we're looking at the long reads, on the other hand, we might just have it that way. That's all we have. We have a representation for a sequence that corresponds to transcript 1, another sequence for several that correspond to transcript 2 and transcript 3. So they just give us a very clear picture. And you might say, problem solved, all is good. We just take the long RNA sequences and straight from the sequencer, we map them to the genome, and voila, that's our gene. Turns out that it's really not all that good, because you'd expect it to look like that, but in reality, when you map it to the genome, it looks more like this. And you can have many more than, obviously you're going to have multiple copies. So what am I showing you here? We have a large number of sequencing errors. And sequencing errors mean that we're not going to be able to assign very precisely the portions on the genome. Sequencing error. It is particularly problematic close to the splice sites. So this might be an inconvenience. For this read, we can definitely say it belongs to this one, this transcript. We have all the exons and they are fairly accurately resolved. But this one, can we tell what transcript is coming from? It could come from the first one, but it could also come from this one. So we don't really know, it's ambiguous. Okay, so when we're quantitating this gene, where do we put it? We have the problem for short reads, but for short reads we have so many of them that we can deconvolute them with mathematical models. We don't have enough reads here to be able to do that. So we just have to throw them away. And our quantification is a little thrown off. We can have an example like here, so the boundary is not precisely aligned. Another example that I'm showing here is this should actually have had an exon here, but it couldn't recognize the splice sites, and rather it put the sequence at the boundaries of the adjacent exons. So that's another case. And you see a few other examples. So there's you're going to see, if you're viewing them in the integrated genomics viewer, you will see that there are gaps. Because there tend to be blocks that are just inserted. The sequence is the way the DNA and the RNA are being put to the nanopore, to the pore. So what you'd expect them to look very clear, like here you actually have a lot of ambiguity, and you have some artifacts, not all of which can be resolved. So this is still a problem. So as a result, we have a slightly different organization here in a regular pipeline. Yeah, we start with the RNA, and everything that I told you about how you choose your RNA, total versus totally selected, that stays here, so you're creating the libraries. But once you create the libraries, they are no longer fragmented. So they're just being fed through this nanopore and read as they come out. And you have a number of reads for every gene. Many of them are partial, actually more than one would expect, looking at the data. So the next step is to make sense of that kind of data through a pipeline. And the pipeline must necessarily entail mapping to the genome. Those mappings are not going to be entirely accurate, as I've shown you, so there are some sequencing errors. So the next step is to actually correct those. And it's easier to correct things when you have multiple copies of them. So we're going to put as many as we can to get more information and perhaps some signal. So the next one is correct and cluster, so we have more information. We then collapse them. That's also part of quantification, because now we can assign each one of those transcripts or portions of transcripts to a certain representative transcript class. Like the different classes, different transcripts that the genome produces. And finally, once we have this is the same as we would have obtained from next-gen sequencing. So now we have the two sets of samples, the controls, the tests and the controls, and we can use some of the previous differential expression, differential spacing tools that I mentioned earlier, DEC, MinJulip, RMAX, and so on, to do this analysis. But the part that is different is this one here. And there are specialized pipelines to take care of that, to do that. None of which is perfect, because nothing in bioinformatics is ever perfect. This is one example that I found fairly easy to work with. How far is it from the truth? I mean, I found others that sometimes work better. Another one would be isopon. I have that in the Coursera lectures. But this flare line, so it's nice because it has all the stages there. Okay, it starts with the reads. It aligns the reads unless you've done it separately, and it's telling you how to do it. Okay, this is what the reads would look like. So you see these conclusions. These actually turns out to be an intron retention case. So then it does a step of the clustering and correction. So it corrects it by looking at additional pieces of external information, namely gene annotation from GenCode. It says, well, I'm within 20 bases of this splice site. I think I should be at this splice site, and it chooses that. Sometimes it's incorrect. Sometimes it might merge a new splice site and replace it with an incorrect but null one. So that's what you'd expect to get there. Are these true? I don't know. I don't know at this point. So the corrected reads are then grouped by their pattern of junctions. So all the three here have the same junctions. This is by itself and so on. They are being collapsed to create these representative transcripts, which would be the transcripts from the gene, 1, 2, 3, 4 classes. There's another level of filtering, and you really can put filters everywhere here, maybe to see how much support you use from reads. Finally, this filtered set can be quantified, and they simply assign whatever transfers they can after filtering they are here to these classes and to do the differential analysis. So all of that is embedded into that pipeline. I'm not saying that it's the best pipeline, but it's a fairly easy to use pipeline. It also has a component at the end that, so it has embedded tools for doing differential expression, differential splicing, and the differential expression is at the transcript level as well. So fairly easy to use if you want to use something like that. Okay, so I'm going to close it here. There's more on the slides for a practical level. I don't think somebody in the exam is going to ask you about how you do an RNA-seq analysis per se. I mean, look at the tools and so on. If you need help with designing experiments or analysis and you don't find the materials that you have sufficient, come and ask me. Send me an email. Yeah. Yeah, thank you.
You're more confident than I am that won't get asked on a comprehensive exam.
Oh, really? Maybe I should make the slides after they move the slides that I didn't want. No, I can do that. I mean, I can do that for the official thing, and they can have the official so they can refer to them. Yeah.
heard you, she's telling you to take that question off the.
That was my question.
Oh, no.
I love your cunty star.